Hi, welcome to the second part of the videos about lymphatic system. This time we're looking on the section through a lymph node. Lymph nodes are peripheral encapsulated lymphatic organs that are inserted into the lymphatic pathways. They work as small filtering devices. Arteries supply the organs and tissues in human body with oxygenated blood and nutrients. They branch into smaller vessels called capillaries. During this transfer of the nutrients, fluid is filtered from the capillaries to the tissues around. This fluid is now called interstitial fluid and it's drained via lymphatic vessels. These vessels join together into the biggest ones called thoracic duct and right lymphatic duct. Lymph that is transferred into the lymphatic vessels has to be filtered through at least one of the lymph nodes before it can be returned into the blood circulation. Lymph node is covered with a capsule on its surface, which is made of dense irregular connective tissue. Trabecules extend from this capsule and divide the lymph node into compartments. Imagine trabecules as columns. They aren't the same structure as septae that you saw in the video about thymus, although they might look the same. Supporting structure is composed of reticular connective tissue, reticular cells, and reticular fibers, that's collagen type 3. We differentiate convex surface and a hilum. Afferent lymphatics enter the convex surface and bring the lymph from the periphery. Arteries and nerves are coming from the hilum to the node, and veins and efferent lymphatic are leaving. It's really hard to distinguish these size lymphatics from the veins, so don't worry about that too much. No one is going to penalize you on the exam because of that. I hope so. Lymphatic vessels do have endothelial cells, but when they enter the node, they drain into spaces that are partially bounded with reticular fibers and cells, but do not have endothelial cells. These spaces are called sinuses. Let's have a look on the other slide where you can see the biggest sinus, the subcapsular sinus that is right under the capsule. We differentiate also perifollicular or peritrabecular and medullary sinuses, which I'm going to describe later. Thanks to these sinuses, immune cells can be in direct contact with the antigens and lymph, which flow is slowing down in the node. Lymph node is divided into cortex and medulla. Cortex contains follicles, which basically are B lymphocytes arranged into the spherical formation. Not only B lymphocytes, but T helper lymphocytes, macrophages, follicular dendritic cells, which are special antigen-presenting cells, and of course reticular connective tissue can be found here. Follicle is divided into several parts: germinal center, mental zone, and marginal zone. We differentiate primary and secondary follicles. Secondary follicles contain germinal centers because they were stimulated with antigens. Primary follicles weren't stimulated, so they don't have one. These primary follicles are uncommon in a healthy human body, so most of the follicles that look like primary are probably secondary ones, sliced about the germinal center. B lymphocytes, also as T lymphocytes, are made in bone marrow. They travel into the periphery and synthesize their receptors. The major one is BCR, or B cell receptor. This receptor is an immunoglobulin incorporated into the membrane of a B cell. These cells can come in contact with antigen in the lymph node. Antigens are presented on the antigen-presenting cells, like follicular dendritic cells. B lymphocytes start with the secretion of immunoglobulins or antibodies M, but these antibodies aren't as specific, and B cells, in effort to improve their affinity or an ability to bind antigen, go to the germinal center, where they can proliferate and undergo mutations. They differentiate into central blasts, cells with light cytoplasm and light active nucleus. Centroblasts with the best affinity to antigen differentiate into centrocytes, which are no longer dividing. They've got dark nucleus that looks somehow broken because of the deep indentation or a notch of the nucleus. Densely packed centroblasts create a dark zone. Paradoxically, dark zone is a zone of proliferation. Centrocytes, which are sparsely distributed, create light zone of selection. It's really hard to separate these zones on slides, but you can distinguish single cells for sure. The most specific centrocytes are able to differentiate into plasma cells and memory cells. In the consequence of gene recombination, these cells can produce specific antibodies like IgG, IgA, and IgE. In order to produce these antibodies, they have to meet with T helper lymphocytes, which transfer the signals to the B cell. Plasma and memory cells then pass through the mantle zone and marginal zone of the follicle. Memory cells partially cross to the circulation, and plasma cells reach medullary sinuses. So, plasma cells are effector cells of the B lymphocytes. Paracortex is located between the cortex and medulla. This region is time-dependent, so it's basically dependent on time as delivery of the T cells. Medulla is composed of medullary sinuses, which drain into a afferent lymphatic vessel, and medullary cords. Cords are well seen in the first slide of a lymph node, and they are closely packed lymphocytes and plasma cells. Lymph nodes are necessary in process of immune response. They clean up the lymphatic fluid and put it back into blood circulation. Thanks for watching. Hope you enjoyed my video about the lymph node. And don't forget to subscribe to my channel. Third part is going to be about the spleen, so don't miss it. Bye!